We see that how Pasteur worked for the germ theory and how he gave idea about the that how microorganisms are responsible for the diseases in human beings. Now let's talk about the Robert Koch and his postulates. Robert Koch also worked for the germ theory, and he found out the this causative agent of the disease anthrax. So let's talk about it how he did it. So Robert Koch, he was a German physician actually. So Robert Koch, he was a German physician, and he discovered the cause of anthrax in around 1876. So what he did actually, that time in Europe, the disease anthrax was destroying a lot of sheep. That's why he decided to work on it. He examined all the disease organisms. Okay, he examined all the disease organisms, and he took out their blood samples. Observed their blood samples and all. And what he found out, he found a particular organisms, which is generally now known as a Bacillus anthracis, which is nothing but the causative agent of the anthrax. So he found out a particular organism or another entity is present in all the ill animals, but it was absent. It was absent in all the healthy animals. So he observed a lot of diseased animals, and he found out that this particular organism or this particular thing is present in the blood sample of every diseased animal, but it is absent in healthy animals. So he knew that it was not a sufficient reason to conclude that this is the causative agent of the disease. So what he did, he performed an experiment to find out its final answers. So here you can see that this is the blood sample of the diseased animal, and this is the blood sample of healthy animal. So these are the RBCs and the other part of the blood, and here this is the RBC and all. But here you can see there is another entity is also present in the blood sample, which is absent in the healthy animal. So this was the finding of the Robert Koch. Then to prove this finding, he conducted one experiment. So what Robert Koch did actually, he took the sample from the deceased animal, a blood sample from the deceased animal, and inject that sample into a healthy animal. Then what happened? Healthy animal developed the same disease. Then again he performed the same procedure. He took the sample from the deceased animal, inject into a healthy animal, took the sample from the deceased animal and inject into a healthy animal. He repeat that process for the several times, and he come across with the same conclusion or the same result. Then what after that what he did, that he isolated that organism as a pure culture on the petri dish, and again inject that pure culture which he isolated on the petri dish to another healthy animal, and again the healthy animal formed the same disease. So from that he concluded that this organism is nothing but the responsible for the disease in the responsible for this disease anthrax. So from his then apart from finding the causative agent of the anthrax, he also found out the causative agent of the tuberculosis, which is nothing but the Mycobacterium tuberculosis, by performing the same experiment and all. And finally, he gave his conclusion in the form of his postulates. So let's talk about its postulates. So, what are Koch's postulates actually? So, Robert Koch gives some criteria for finding out the causative agent of the disease. For finding out the causative agent of a disease. So, for example, like what is the use of this postulate? So, if you really want to find out causative agent for a particular disease, so and if you have come across with some organism which is present in the the diseased animal, so it should follow all these postulates. Then only you can conclude that the that organism is responsible for that particular disease. So let's see what are these postulates. The first postulate was the same pathogen must be present. Okay, that the same pathogen must be present in all cases. From the first postulate, Robert Koch said that if you're observing a particular organisms which has formed a particular disease, if you have observed a particular organism in its samples or in its body, so it should be present in all the cases. For example, like if you have studied one organism, and you have come across with with a certain different organism which is generally not present in the healthy animal but in diseased animal. So it should be present in the another animal which is suffering from the same disease. If you have studied the 50 animals which are suffering from suffering from a particular disease, so that organism should be present, or that pathogen should be present in all those 50 animals. If you are studying the 100 animals which are suffering from the disease, that pathogen should be present in all those 100 animals. So that is the simply a simple meaning of the first postulate. Then second postulate is that the organism must be isolated and grown in pure culture form. So this is one of the very important criteria, that if you have found out that this particular pathogen is present in all the cases, and you have kind of thinking that this is responsible for the disease, so you should be able to isolate that organism in a pure culture form, that is on the petri dish. Then so upon inoculation of this pure culture or the isolated cells, it should cause the same disease again, it should form the same disease in another healthy animal. So, for example, like if you have performed an experiment to identify the causative agent, and you have, you have found that it causes the same disease in, it is present in all the cases, and you have isolated it. So, upon inoculation of that pure culture into an another healthy animal, so it that healthy animal should be able to form that, sorry, not that pathogen should be able to cause the disease in the healthy animal, same disease in the healthy animal. Okay. And the last postulate was that pathogen must be re-isolated from the inoculated animal. So you have observed one disease animal, you have taken out its sample, you have isolated that pathogen in the form of pure culture from that diseased organism. Then again you have inoculated that pure culture into another healthy animal, and now you should be able to re-isolate that pathogen from this healthy animal. Okay. So you should be able to re-isolate those that organism from the that healthy animal to which you have inoculated that pure culture cells. So these are the four postulates of the Robert Koch. And if a particular organism is responsible for a particular disease, so it should obey all these four postulates. But of course, there are certain cases which shows some exceptions for this Koch's postulates. For example, there are some microorganisms which cannot be grow as pure culture. Example of this such a microorganism is the Mycobacterium leprae, which causes leprosy in human beings, and Treponema pallidum, which causes syphilis, and the Treponema pallidum, which causes syphilis. So there are certain other bacteria or the other microorganisms as well which could not be grow or which could not be isolated on the petri dish or on an artificial media. So this is the exception to the Koch's postulates. Then in some experimental animal, then in some cases experimental animal for inoculation is also not available. For example, you can take an example of cholera. Cholera is present. Cholera, is caused by the Vibrio cholerae, which causes a cholera in human beings. But you could not inoculate that organism into human beings for your experiment purpose. So, for certain cases or for certain diseases, for certain organisms, you don't get an experimental host suitable experimental host to perform your experiment. So, in that way, there are certain exceptions to Robert Koch's postulates, but in maximum cases, generally the microorganism follows all the postulates. But there are certain exceptions which generally don't come under the category of these postulates. That's it for today's video, and thank you so much for watching.